Hello everyone, welcome to Merit. So under biochemistry, we are going to talk about transport system. And this topic will cover the transport of molecules, okay? So we are going to talk about molecular transport. And since this transport will be across membrane, it will be also called as membrane transport. So in this video, we'll talk about the classification of molecular transport and its types, and lastly, some examples. So on the basis of number of particles, or let's say types of particle transfer at a single time, we classify transport system as uniport and co-transport. So uni means single, okay? So single transport of particles occurring at the single time will be uniport. And the next is co-transport. It is of two types, okay? Here two particles get transferred at a single time. This transfer can be in the same direction or in opposite direction. So in the same direction, it will be called as symport, and in opposite direction, it will be called as antiport. So example of symport includes sodium dependent glucose transport. An example of antiport will be sodium pump and chloride bicarbonate pump. Okay, this sodium pump is responsible for nerve conduction. You might have heard about the action potential things. Okay, here the sodium pumps acts, and the second is from Haldane effect, or also called as chloride shift. This chloride bicarbonate pump is responsible in chloride shift. Okay. So on the basis of energy dependency, it is of two types, passive and active transfer. So passive means requiring no energy, and active means requiring energy. So passive transfer, there are three types of passive transfer, that is simple diffusion, facilitated diffusion, and ion channels. So simple diffusion. Since it is a passive process, there is no ATP usage, and the process occurs along the concentration gradient, meaning from higher concentration to lower concentration. And it will occur till the equilibrium is achieved, which means the concentration on both sides should be equal at the end. And the rate will be directly proportional to the concentration gradient. More the concentration gradient, faster the diffusion occurs. There are other factors too which governs the rate of the diffusion, and this includes the hydrophobic character of the molecule and the chargeless particle. Chargeless particle and hydrophobic molecules diffuse faster than the hydrophilic molecule and charged particle. Being a passive process, it is a slow process. An example includes diffusion of gases such as oxygen, carbon dioxide, carbon monoxide across the membrane. The next is facilitated diffusion. It is a carrier-mediated process, which means it requires a carrier molecule. And since it requires a carrier molecule, the process is faster than the simple diffusion. The next property is it follows the saturation kinetics. Now just remember two things, because we are going to talk about the saturation kinetics in the future chapter. We just need to remember two things now. The first thing is rate depends upon occupancy of active sites of the carrier protein, and it can be inhibited by simple compounds which can bind to the active site. So similar looking substance can bind to the active site and hampers the process, which means it can inhibit. Okay, so facilitated diffusion can occur bidirectionally, which means it can occur in both directions, okay? And these carrier molecules occurs in two states, either pink state or pong state. In pong state, the active sites are exposed to the exterior, and higher concentration of the solute in exterior will result binding of solute at the active sites. And this binding will cause conformational change in the molecule, which bring the solute inside, and carrier protein comes in pink state. Okay, and in pink state, the active sites are facing in the inner wall of the membrane, where concentration is minimum, and this minimum concentration will result detachment of the molecule from the active sites, and hence the process occurs again and again. Example includes fructose transfer. It can be easily remembered F for F, facilitated diffusion and fructose transfer. The next is ion channels. These channels are responsible for ion movement, and these are nothing but transmembrane protein, which means the protein will be throughout the membrane, from outer membrane to the inner membrane. There is a quick transport of ions which is usually acting on fractions of seconds, which is very fast, and these channels only allows specific ions through them, which means these channels are selective to particular ions. These channels can be either gated or non-gated. They open when there is a stimulus. When a stimulus arrives, there is rapid influx of ions, and being a passive process, it occurs along the concentration gradient. And they are usually associated with nerve conduction, secretions, and synaptic transmission. Okay? So gated ion channels, they are either ligand gated or voltage gated. Ligand is a molecule which binds to a specific receptor. This acts as a chemical stimulus. It can be either intracellular or extracellular. One of the most popular examples is acetylcholine receptor. Here acetylcholine binds to the postsynaptic terminal. And here, this on binding opens the ligand-gated sodium channels. This causes depolarization of the membrane and nerve conduction occurs. These channels are open till acetylcholine is bound. So it needs to be degraded as soon as possible. So to close the channel. And cholinesterase are the enzymes that degrade the acetylcholine, so that acetylcholine rapidly degrades, and hence these channels open only for a brief milliseconds. The next is voltage gated channels. They open when the membrane depolarizes, and this causes change in voltage across the membrane. And after reaching a threshold voltage, the channels open. And when it opens, rapid influx of ions along the concentration gradient occurs. It is also involved in nerve conduction. The next is the active process, which means requiring energy, and it requires energy in the form of ATP. This process is unidirectional process, unlike passive, which can be both bidirectional or unidirectional. It occurs through transfer proteins and also follows saturation kinetics, which means similar compound can occupy the active sites and can inhibit the process. Now the examples of active transport includes sodium pump and calcium pump. Sodium pump is the best example because it is found in almost every cell, and it is also a antiport system, meaning each transfer will include transfer of 3 sodium and 2 potassium in opposite direction. The second example is calcium pump. It is responsible in muscle contraction, and its function is to maintain low cytosolic calcium. Keeping low is necessary so that the muscle gets relaxed for next contraction. So relaxation of muscle is also an active process, meaning ATP will be used for muscle relaxation. And each ATP will cause transport of 2 calcium molecules or ions. So what are the difference between ion channels and ion pumps? So in ion channels, there is movement along the concentration gradient and the process is passive, while ion pumps cause movement of ions against the concentration gradient. Ionophores. They are responsible for increasing the permeability of the cell membrane and is essential for transport of some antibiotics. They can either be mobile ion carriers, as in valinomycin transport, or channel formers, as in gramicidin. So AQUAPORINS, these are nothing but water channels, and these are for movement of water molecules. There are 10 types of water channels, and mutation in these channels can cause diabetes insipidus. So let's summarize. In simple diffusion, there is no carrier protein, it is towards the concentration gradient, there is no energy expenditure, and the examples are gaseous transfer across the membrane. The next is facilitated diffusion. Here there is a carrier protein, however there is no movement against the concentration gradient and there is no energy expenditure. The example includes fructose and glucose transfer. In primary active transfer, there is a carrier protein and the transfer occurs against the concentration gradient, there is direct expenditure of energy in the form of ATP, and the example includes sodium pump. In secondary active transport, there is a carrier protein. It is also against the concentration gradient. However, the energy expenditure is indirect. The example includes glucose transport in intestine. And in ion channels, there is a carrier protein. It is not against the concentration gradient, but towards the concentration gradient. And there is no energy expenditure. And the example includes sodium channel. Thank you for watching this video. Do subscribe for more. This is Medit signing off. Medit for you.